My dear students, welcome again in our lesson. Remember before break, we had a question requiring you to describe the mechanism of carbon dioxide fixation in sugarcane. Describe the mechanism of carbon dioxide fixation in a sugarcane plant. I did say that the question of concern that you should ask yourself before going into finding the answers is why a sugarcane plant? What is a sugarcane plant? And I gave you a clue that you have to remember the examples, the representative members of the C3 plants and C4 plants. Now I think you have come to realize that a sugarcane plant is a representative of the C4 plants. And therefore, the requirements of the question is that you describe the mechanism of carbon dioxide fixation in the C4 plants. We have seen that it is involving fixation of carbon dioxide in the mesophyll cells, shunting of malic acid from mesophyll cells to bundle sheath, chloroplasts, regeneration of phosphoenopyruvic acid in the bundle sheaths, and refixation of carbon dioxide in the bundle sheaths. Now, having seen that, let us move on to the factors that affect the rate of photosynthesis. Factors affecting well, my dear student, there are several factors that influence the rate at which photosynthesis occurs, that affect the rate at which photosynthesis takes place. And these factors can be categorized into A, external factors, and B, internal factors. External factors in relation to the plant, and therefore these are referred to sometimes as environmental factors. And the internal factors relative to the plant, these are also referred to as plant factors. Now, before we see these factors, let us look at the concept of limiting factor. Now, what is a limiting factor? Suppose that you have a chemical process, and this chemical process is involving several steps. And these several steps are taking place at different rates. The slowest step in the series of these reactions will determine the overall rate of that chemical process. And this slowest step is the one which will act as a limiting step. Now, if we go into a physiological process which is involving several factors, a physiological process whose proceeding requires several factors, that the factor which will be in a shortest supply will limit that process, and therefore it will act as a limiting factor. For example, consider a photosynthesizing plant. For photosynthesis to take place, there are several factors, there are several requirements, such as carbon dioxide, photosynthesis requires water, photosynthesis requires light, Photosynthesis requires enzymes and the likes. Now, suppose that you have this photosynthesizing plant. If all other factors are normal, if all other factors are normally supplied, this plant has a plenty of carbon dioxide, there is water, enzymes are efficient, temperature is normal, but this plant is devoid of light. Light is absent. Automatically, this plant will not photosynthesize. And therefore, light is acting as a limiting factor. If light is available, maybe it's during the day, light is available, the enzymes are active, water is there, but this plant is lacking carbon dioxide. The plant will not photosynthesize, and therefore carbon dioxide will be acting as a limiting factor in that way. Therefore, the principle of limiting factors, that when a chemical process is determined by several factors, that the factor which is near its shortest supply will act as a limiting factor. That the factor which is near to its shortest supply is the one which will act as a limiting factor. Now, having seen the concept of limiting factors, let us go to the external factors. The external factors, or simply environmental factors, these are of different sorts. The first one talk of sunlight, light intensity. Well, light has an important role in the process of photosynthesis. You can remember what we said when we were discussing, the light-dependent phase of photosynthesis. We saw precisely the role of light in that particular phase. And therefore, initially, the rate of photosynthesis will vary proportionally with the intensity of light. With increased light intensity, the rate of photosynthesis is also increasing. However, it reaches a point. This is called a light saturation point, where the rate of photosynthesis tails off and attains a constancy. At this stage, when the plant has reached its light saturation point, any additional light intensity will not result into increased rate of photosynthesis. However, in some plants, above additional light above the light saturation point will result into increased rate of photosynthesis, provided that the concentration of carbon dioxide is high. What I'm saying here is consider this graphical illustration. So it means the rate of photosynthesis goes proportionally with increased light intensity. However, at a certain point that we are going to call light saturation point, the rate of photosynthesis tails off. This is a light saturation point. And therefore, it will be constant. However, in some plants, with increased concentration of carbon dioxide, the rate of photosynthesis will increase, meaning that here we have low carbon dioxide concentration. Here we have high carbon dioxide concentration. Also, remember that while the plant is photosynthesizing during the day, there is another physiological process that is taking place, that is respiration. And these two physiological processes are taking place in two different compartments. Photosynthesis taking place in the chloroplast, and respiration taking place in the mitochondrion. While photosynthesis is giving out oxygen as a waste product, respiration gives out carbon dioxide as a waste product as well. Now, there is a certain light intensity, during which the two processes become equal. We know, under normal circumstances, and we were told so, that during the day, the plant takes in carbon dioxide and gives out oxygen. Well, practically it is true. And this is because during the day, the rate of photosynthesis is always higher than the rate of respiration. And therefore, the amount of oxygen given out as a result of respiration exceeds the amount of carbon dioxide that has to be given out as a result of respiration. And in addition, with the carbon dioxide taken from the atmosphere, the plant also utilizes the carbon dioxide given out as a result of respiration. And therefore, the net atmospheric gain of air is oxygen. And this is the essence of saying that during the day, the plant gives out oxygen and takes in carbon dioxide. Now, at a certain light intensity, the rate of respiration equals the rate of photosynthesis. So that the amount of oxygen given out during photosynthesis will be equal to the amount of carbon dioxide to be taken in during photosynthesis. Consequently, the plant will utilize that oxygen from photosynthesis for respiration, and the carbon dioxide from respiration for photosynthesis, so that the amount of carbon dioxide to be given out as a result of respiration is equal to the amount of carbon dioxide to be utilized during photosynthesis. And therefore, the net gain of air from the plant will be zero. And that will be called a compensation point of a plant. Here we have compensation point of that particular plant. And therefore, net gain of air will be equal to zero. Here we have a zero mark, and here we have carbon dioxide taken in, and here we have carbon dioxide given out. That is the general idea, the concept of light as far as photosynthesis is concerned. Now, let's start the question. That relate light intensity to photosynthesis. Relate light intensity to photosynthesis. How the intensity of light affects photosynthesis. How the intensity of light affects photosynthesis. So apart from light intensity, the second factor that may affect the rate of photosynthesis is carbon dioxide concentration. Well, the atmospheric carbon dioxide has a concentration of roughly 0.03%. And therefore, the rate of photosynthesis will increase as a result of increasing concentration of carbon dioxide. until a maximum of about 0.1% is attained. Above this concentration, carbon dioxide will damage the plant leaves and the photosynthetic failure will be experienced. So the concentration of carbon dioxide as it increases from 0.03%, the rate of photosynthesis is also increasing. However, above 0.1%, the concentration of carbon dioxide will have a detrimental effect to the plant and the photosynthesis will cease altogether. Another factor, the third one, that is temperature. The light-independent phase of photosynthesis. The light-independent phase of photosynthesis is controlled by enzymes. And therefore, it is temperature dependent. You can remember from biochemical point of view that the rate of enzyme action is increasing with increasing temperature. So as the temperature increases, so does the rate of photosynthesis. The optimum temperature for the temperate plants for photosynthesis to operate is 25%. 25 degrees centigrade. However, according to the Q10 rule, from 25% to 35%, the rate of photosynthesis doubles for every 10 degrees centigrade rise in temperature. The Q10 rule states that the rate of a physiological process doubles with every 10 degrees centigrade rise in temperature, provided that the temperature is within the range that can be tolerated by a living system. The rate of a physiological process will double for every 10 degrees centigrade rise in temperature, provided that the temperature is within the range that can be tolerated by the living system. For example, suppose that rate at 5 degrees centigrade is R. What will be the rate at 15 degrees centigrade? If the rate of a physiological process at 5 degrees centigrade is R, and the rate of the same reaction, the same process at 15 degrees centigrade, remember there is an addition of 10 degrees centigrade, and we have said according to the Q10 rule that the rate of any physiological process will double with every 10 degrees centigrade rise in temperature, provided that the temperature is within the range that will be tolerated, that can be tolerated by a living system. So the rate at 15 degrees centigrade will double. That will be 2R. Now what will be the Q10? This will be rate at 15 degrees centigrade over rate at 5 degrees centigrade. And this will be equal to 2R over R. And this will be equal to 2. This is what we mean by double. Rate of a reaction doubling goes twice as much with even 10 degrees centigrade rise in temperature. So the optimum temperature for the temperate plant, we have said that it is 25 degrees centigrade, although others go as far as 35 degrees centigrade. Above this temperature, the photosynthetic enzymes will undergo denaturation, and therefore photosynthesis will cease. And below this optimum temperature, the enzyme becomes inactivated and the rate of photosynthesis goes low. Apart from that, we have the fourth factor, This is water. Water is the fourth external factor which determine the rate or which affects the rate of photosynthesis. We know from light reactions of photosynthesis that the role of water is to provide hydrogen ions and electrons from photolysis. Remember photolysis of water, a photochemical splitting of water in the presence of sunlight. This will give hydrogen protons, a pair of electrons, and oxygen. What is the role of these electrons in the light reactions? What is the precise role of the hydrogen protons in the light reactions? Try to remember what we said. Now, apart from being a source of hydrogen and electrons, water also plays an important role in the translocation of materials in the plant, such as gases, such as mineral salts, and the products of photosynthesis, I mean the food materials. Lack of water results into wilting of the plant leaves. And when the plant leaves wilty, the consequence of this is a closure of stomatopores. When the stomatopores are closed, then there will be reduced, if not minimized, intake of carbon dioxide, and consequently photosynthesis will fail. So you can see that water is important in the life of the plant, photosynthesis being included. Although the amount of water required for photosynthesis per se is very, very minimum, very, very small. However, the plant should not be deprived of this small amount of water, otherwise no photosynthesis will take place. The fifth environmental factor which affects the rate of photosynthesis, we have mineral salts. Mineral salts that we are talking about here include magnesium and nitrogen. Although these do not have a directly influence to photosynthesis, but their absence will lead into photosynthesis failure because they are integral parts of the chlorophyll. These are important inorganic ions in the structural makeup of a chlorophyll. And therefore, their absence will result into a plant being chlorotic. The leaves become yellowish or somewhat whitish due to lack of these two elements, due to lack of these two minerals. And absence or lack of chlorophyll means that the plant will fail to absorb sunlight. And when the plant fails to absorb sunlight, automatically no photosynthesis will take place. Those were the 5 factors, external factors, that determine the rate of photosynthesis. Second one, we have internal factors. We have the internal factors. Internal factors, or simply plant factors. These are called plant factors because they are related to the structure and the physiology of the plant. The first one, we have chlorophyll concentration. Chlorophyll concentration. We know precisely that the role of chlorophyll is to trap light. The greater the concentration, or the higher the concentration of chlorophyll, the greater will be the amount of light that will be trapped into the plant. And automatically, the higher will be the rate of photosynthesis. So with reduced concentration of chlorophyll, the rate of photosynthesis also goes down. Now, what factors that can lead into reduced concentration of carbon dioxide? One, fungal and bacterial diseases. Two, it can be due to lack of mineral salts, such as magnesium and nitrogen, which I have said that they are integral parts of this chlorophyll. So either due to lack of those mineral nutrients, or due to fungal and the bacterial diseases, the amount of carbon dioxide, the amount of chlorophyll will go down. When the amount of this chlorophyll goes down, automatically the rate at which photosynthesis is proceeding goes down as well. Secondly, we have leaf. I mean leaf size, shape, or morphology. Well, the general structure of the leaf has also a great influence to photosynthesis. We have broad leaves. If the leaf is broad, it is much exposed to light, more exposed to carbon dioxide. It will take in more light, it will take in more carbon dioxide, and therefore the rate of photosynthesis will be high as compared to the narrow. However, the leaf, which is thin, has a shorter distance across which carbon dioxide and light will have to travel. And therefore, in the thin leaves, the rate of photosynthesis is higher compared to the thick ones. Also, if the leaf has a thick cuticle over its surface, in a bid to resist excessive loss of water, the amount of carbon dioxide that will be diffusing in automatically will be small, being limited by a thick cuticle, and therefore the rate of photosynthesis indoors and in such like a plant with such like leaves will be smaller compared to the plant whose leaves have a thin cuticle. So the third internal factor that affects the rate of photosynthesis, we have enzymes. Photosynthesis is controlled by 3 different enzymes. We have ribulose biphosphate carboxylase of the C3 and the C4 plants. We have phosphoenol pyruvic acid carboxylase of the C4 plants only, but we also have ATP synthase, which is used to catalyze the synthesis of ATP in the light reactions. All these enzymes work under optimum conditions of temperature. When the temperature is high, or when the temperature is higher than the optimum level, these enzymes will be denatured and the photosynthesis will fail. And when the temperature is lower than the optimum, the enzymes will be deactivated, will be inactivated, and therefore the rate of photosynthesis again may be slowed down or may cease altogether. So this is how enzymes affect the rate at which photosynthesis is taking place. The fourth The last but not least, we have inhibitors. We have inhibitors. An inhibitor is any factor which may hinder the development or formation of a given or continuity of a given process. Now, the rate of photosynthesis can be inhibited by various factors, but the most inhibitors of photosynthesis are the herbicides. We have the herbicides. Example of the herbicides that hinder photosynthesis. We have chlorophenyl. We have chlorophenyl dimethylurea. Chlorophenyl dimethylurea, which normally interferes with electron transport process during the rate-dependent phase of photosynthesis, and because of interference with the electron transport, then there will be no photophosphorylation. No photophosphorylation, and therefore no ATP will be formed, and therefore the process of photosynthesis as a whole is inhibited. But also, apart from the herbicides, we have oxygen. High oxygen concentration will inhibit photosynthesis, especially in the C3 plants. We have seen that these C3 plants are using ribulose biphosphate carboxylase enzyme, which is competitively inhibited by oxygen. So with high concentration of carbon dioxide, photosynthesis in the C4 plants is inhibited. My dear student, remember, what we have at hand is autotrophic nutrition. We started by defining what autotrophic nutrition is. We went as far as photosynthesis, chemosynthesis, mechanism of photosynthesis, light-dependent phase, dark-dependent phase, light-independent phase of photosynthesis, differences between C3 and the C4 plants, including their meaning. We have seen the mechanism of carbon dioxide in the C4 plants, and now the factors that affect the rate of photosynthesis. We have come to an end of autotrophic nutrition, especially photosynthesis. Before we conclude, here are the study questions. Describe how C4 plants. Describe how C4 plants are adapted to synthesize sugar in tropical and subtropical climates. Describe how the C4 plants are adapted to synthesize sugars in the tropical and the subtropical climates. The second question, using your knowledge, on C3 and the C4 plants. Explain which plants are more vulnerable to climate changes. Or in other words, using your knowledge on C3 and the C4 plants, explain which plants are more vulnerable to damage due to climate changes. The third one, explain how the following affect the rate of photosynthesis. 1, light intensity. 2, enzymes. And 3, inhibitors. While you are trying to seek for the answers on these study questions, you may consult, apart from the notes you have been taking during this session, you may also consult biological science, by Taylor. You may consult Understanding Biology for Advanced Level Students by Susan Toole and Glenn Toole. Understanding Biology. But more importantly, I'm stressing on this. Consult Biology for Advanced Level Students. Biology for Advanced Level Students. Book 5, or book Form 5. A book which was or which has been currently, a book that is currently issued by the Tanzania Institute of Education. You may miss biological science, you may miss Understanding Biology, but please don't dare to miss this book, which has been currently issued by the Tanzania Institute of Education. My dear students, having seen this, we have now come to an end of today's session. Remember, in our next meeting, we shall start discussing nutrition in the plants. It will be an exciting discussion. It will be an exciting session. So please remember to come close to your television set when the next session comes in. My name once again is Fortunatus Kiobia. Bye-bye.